right, continuing on with Saturday's study session review for microevolution, let's look at question number 6. And question number 6 is asking us, these researchers studied genetic variation in the muscle Mytilus edulis. I was once given the advice that if you come across a name you're not sure what it is in AP Biology, because they're going to throw lots of words out at you that you may or may not know what they are. This is a scientific name. To get your brain in the right frame of mind, it's okay to replace anything you don't know with the words Mickey Mouse. So this marine mussel Mickey Mouse in Long Island, New York, they measured the frequency of the LAP94 allele that codes for a protein involved in internal water balance, are some of the key words there. It says, which of the following is the best explanation for the differences in LAP94 allele frequencies? Let's go over to the graph and look at what we see. We see there are the sampling sites 1 through 11, and they start down here. And it also shows us that the frequency of the allele in each of those sampling sites, and you can see it increases as those increase as well. The other thing we need to look at is we're seeing the salinity increases as we go towards the open ocean. So one thing we can deduce from this is that as salinity increases, the frequency of the allele increases as well. So I'm going to put this called LAP94. Let's use that as we analyze some of these questions. First one says, mussels with a high frequency of the LAP94 allele are selected against high salinity areas, and that's not what this is showing us down here. They'd actually be selected for high salinity areas. Next one says, mussels living in areas with lower salinity do not need to maintain osmotic homeostasis. Everything needs homeostasis, and especially if it's a marine organism, it's going to have to be osmotic homeostasis. I'm going to get rid of that one. Excuse me. Mussels with a LAP94 allele are able to maintain osmotic balance in water with high salt concentration. The higher the allele, the higher the salt concentration. So I do like this one. We'll come back to it. High frequencies of the allele increase genetic variation in mussels. And we're not looking at genetic variation in this, so I'm going to get rid of this as well. Go back and let's let our correct answer be C on this one. That's question number 6. Moving on, this is just showing us, reminding us, genetic changes that enhance survival and reproduction can be selected by environmental conditions. This is also the point where I like to remind my students, genes inside you, your genotype, ultimately determines your phenotype, what you look like, and then that can be acted upon by the environment or natural selection. It can choose which phenotypes are going to best survive or not, but what we don't see happening is this idea of the environment choosing the genotype. That's a common misconception. The environment acts upon the phenotype. The phenotype is determined by the genotype, but it's not like the environment is changing the genes directly. So make sure you keep that in mind. Let's go on to the next one, and it's question 7. We're looking at the black-bellied seed cracker finches of Cameroon, and it tells us up here, it says the small beak finches feed mainly on soft seeds. The large-billed finches are for the hard seeds. So we have a connection. I just want to make sure I don't lose that. We've got the small bills are going to eat the soft seeds. The large bills stick with the hard seeds. This graph is showing this connection. Just from the graph, we can see here's the number of birds over here. The width of the bill tends to be, right now, tends to be fairly small width compared to a large width. But the question says, suppose soft seeds become scarce. So we know we have soft seeds versus hard seeds, and the soft seeds become scarce. If I were to make that analogy over here, I'd say, okay, the soft seeds become scarce. What would I deduce from that? Then I would expect the small beaks to become less numerous, the small beak birds to become less numerous as well. It said, what will most likely occur in the beak size of the black-bellied finch population? It says the proportion of birds with intermediate-sized beaks will increase. And really, we've been talking about small or large-sized beaks, not the intermediate size. So I'm going to say probably not that one. The proportion of large-billed finches will increase due to directional selection. Now, if you think about it, if the small beaked birds are declining, then we'd probably see, if the only food source are hard shells or hard seeds, we would probably see a larger population of large-beaked birds. So I'll put maybe next to that one. This one says the beak range will become wider due to disruptive selection, meaning the range you get a greater selection of all of these, and I don't really like that one. And the last one says the proportion of large, intermediate, and small billed finches would stay the same. Not if the food source is changing. That's not going to happen either. So I'm going to lean with B for that one as well. Something just to quickly remember, types of natural selection here. I show my students a video about the rock pocket mice in Arizona desert, where there was a volcanic flow at one time. It changed the desert landscape from just brown to kind of a black and brown. And what we find is researchers were able to find evidence of how the genes have changed inside the mouse to make them more advantageous to live on the black rock, is to have a black color to be hidden from predators. This is an example of directional selection. I asked my students what type of environment would favor disruptive selection, where you get mostly light-colored mice and dark-colored mice, not in the middle. And you could come up with something like maybe it's a white sand beach with a volcano, maybe it's a snowy area with a volcanic rock that these both populations could survive in. And then what type of environment would allow just a medium-colored mouse, a tan-colored mice to survive in, and that'd be like a plain old desert or a beach of some type. And again, this would be stabilizing. The original population looks like this, but we're selecting, or I should say nature's choosing, selecting this phenotype to be the most advantageous. The other ones would be being picked off fairly easily. So let's look at our math gridding question here. This is tells us right here, in population is Hardy-Weinberg equilibrium. The frequencies of the recessive homozygote genotype of a certain trait is 0.09. Calculate the frequency of individuals homozygous for this dominant allele to the nearest 100th. So let's analyze what we have here. This tells us right here the frequency of a recessive homozygous genotype is 0.09. I like to review with my students what do all the different symbols in the Hardy-Weinberg equation mean. And so first thing I'll ask is, what does P mean? And if you don't remember, in case you forget, you can always, this is a great hint, go to your equation sheet, and on the equation sheet what you'll find, excuse me, go back here, is it tells you right here, P is the frequency of the dominant allele. Q is the frequency of the recessive allele. I know a lot of times my students get the terms allele and genotype mixed up, but the study guide tells you right here, the equation sheet tells us that this is the allele. So then from that I could remember that, oh, P squared must be, P was still dominant. This is going to be homozygous recessive genotype. Q squared would be the homozygous, P squared would be the homozygous dominant genotype. Q squared would be homozygous recessive genotype, and this would be our heterozygous genotype, kind of what they look like. So let's go back to that question. We know that P is the allele, and this one's asking us for individual homozygous dominant frequency of the homozygous dominant allele. And basically what that's looking for is P, the homozygous dominant allele. Dominant allele. You can't forget that that's also talking about the frequency of that. So this one's asking us to solve for P, but let's look at what we have. It says for this one there is a recessive homozygote genotype. Okay, so now remember genotype, recessive genotype, we're looking at Q squared, and they give us that value. So I know down here that my Q squared value is 0.09. The way I tell my students to approach these problems is mind your P's and Q's, solve for Q. Once you solve for Q, everything else, it can be fairly easy. So this one gives me my Q squared. How do I get Q from Q squared? Well, Q is the square root of Q squared. So if I plug in that number, this would be the square root of 0.09. And if I have my calculator, what I could do is say Square root of 0.09 is equal to, try that one more time, 0.09 square root, 0.3. So Q is equal to 0.3. Once you solve for Q, everything else is easy because it's really just plug and chug. You have two equations. Let's go back here. Excuse me, let's look at this one. I could look at this one I refer to as the Hardy-Weinberg equation. This one is the easy-Weinberg equation. And when I go back, I remember that I'm just trying to solve for Q in this one. Excuse me, I'm trying to solve for P. And since I already have Q, I'm going to use the equation that says P, the easy Weinberg equation, P plus Q equals 1. If I would like to get solve for P, I already know what Q is, so it's basically 1 minus Q would give me what my P value is. Let's go ahead and plug those in. So 1 minus 0.3 should give me 0.7 for my dominant allele. All right, since I threw this problem in here, we did get the dominant alleles P. It says go to the nearest. Always go back and reread math questions, and they tell you how far to go. This is the nearest 100th. We ended up right here with 0.7. So to make this truly accurate, I would have to put 0.70 as my final answer for P, which represents, again, the frequency of the dominant allele, which also is stated on your equations and formula sheet. So I hope that was helpful for the multiple choice and the grid, the first grid in problem. Next video, what I'd like to look at are the free response questions, and we'll do that here shortly. I hope that this was helpful.